is Adam Cuker. I am a hematologist at the University of Pennsylvania in Philadelphia. And on behalf of my co-authors, I am delighted to provide this abbreviated presentation on our study, Efficacy and Safety of Fidenacogene elparvivec, hereafter referred to as FE, in adults with moderately severe to severe hemophilia B, the Phase 3 BENEGENE-2 Gene Therapy Trial. So by way of introduction, FE is an adeno-associated virus-based gene therapy in development for the treatment of individuals with hemophilia B. It contains a liver-specific promoter and encodes a high-activity variant of human factor IX called factor IX Padua. BENEGENE-2 is a single-arm, multi-site, pivotal Phase 3 study designed to assess the efficacy and safety of FE. All study participants were required to complete a minimum of 6 months of factor IX prophylaxis therapy as part of the lead-in study, after which eligible subjects rolled over to BENEGENE-2. In BENEGENE-2, these subjects received a single intravenous dose of FE at 5 times 10 to the 11th vector genomes per kilogram, and follow-up post-infusion will last for 6 years to monitor long-term safety and efficacy. The primary endpoint for our study is total ABR. That includes treated and untreated bleeds, and treatment with FE was associated with a 71% reduction in total ABR compared to the prophylaxis period, going from 4.4 in the prophylaxis period to 1.3 following gene therapy. This met criteria for both non-inferiority and superiority of FE compared with prophylaxis. And in fact, about 2/3 of subjects had no bleeding events between week 12 and month 15 after infusion of FE. A key secondary endpoint was the annualized infusion rate. Treatment with FE was associated with a 92% reduction in the annualized infusion rate compared with the pre-infusion prophylaxis period. Another key secondary endpoint was factor IX activity levels. These are data that I'm going to quote for you using the one-stage factor IX assay with a synthisil reagent, where between weeks 12 and month 24, mean factor IX levels range between 25 and 30% and remain stable over the course of those 24 months. What about safety? Well, 38 subjects experienced 45 adverse events. There were no adverse events of thrombosis, inhibitor development, or malignancy. The most common adverse event by far was an increase in transaminases occurring in 24 subjects. This led to a high use of corticosteroids. 62.2% of subjects received at least one dose of corticosteroids for a presumed cellular response. So the key take-home messages are: first, compared with prophylaxis, treatment with FE reduced the total ABR by 71% and reduced the annualized infusion rate by 92%. Second, mean factor IX levels were 25 to 30% and were stable for up to 24 months post-treatment. And finally, FE was generally well tolerated with an acceptable safety profile. I want to conclude by thanking all of the study participants as well as my co-investigators and site staff, and noting that this study was funded by Pfizer. Thank you very much for your attention.